Hello, my name is Andy Bukle. I am the research lead in Cardiobonos team at Indiana University. And in this video, I will give you a VR demonstration about senescence hallmarks over a ZenNet dataset with a voiceover by Dr. Himali Fandani. Thank you.
So what we're looking at here, let me talk you through this grid space. Currently, it just looks like a rectangle with a lot of spots in it and a bunch of colored lines sticking up and down. But what it really is a series of spatially resolved gene expression measurements from a slice of the human dorsolateral prefrontal cortex. Each dot, each white circle, represents a 55 micron spot that's going to capture gene expression from anywhere from 1 to 20 cells, depending on the size of the cells. Now, because this is the brain, there's all kinds of different cells that range in sizes. So what we wanted to see is how does the level of expression of known aging-related genes or senescence-associated gene signatures, how does the expression of those change over space and time? So we collated sets of genes that represent these different aging-related or senescence-related signatures, and you can see those are the colors that are shown on here. So there's the San Diego TMC, which is a manually curated set of genes from the San Diego Tissue Mapping Center, which is also part of the SENNet consortium. The blue represents genes that are involved in resistance to apoptosis. The purple is activated p53 targets, and so on and so forth. So each color is a different set of curated genes. Now, sometimes you'll see multiple colors in each spot. Sometimes you'll see spots are enriched in only a few of these. And all of that is information that we can use to understand how senescence-related gene signatures are changing across the landscape of this tissue section. So if you can take a closer look at some of these spots, you'll see that you usually have more than one line. A lot of the spots have several different lines associated with them. Some are above the plane of the spot, some are below the plane of the spot. So if you look at this one, yeah, there you go, see? And the ones that are above the plane of the spot shows that these gene signatures are elevated in that spot with respect to the rest of the spots. The ones that are below the plane of the spot are telling you that those gene signatures are lower and in that spot. So genes are either enriched or depleted in that spot, depending on whether this line is above or below the plane of the section. So now we can start to see how many different colors are associated with a given spot. Are particular colors more likely to be together than others? When one set of colors is up, is there another set of colors that's down? And by doing analyses such as these, we can begin to understand what is happening to the landscape of senescence-associated gene expression changes overlaid onto this landscape of the tissue section. And by seeing it this way, it's a really evocative way of appreciating how much information you can get from an experiment like this. And it brings the data to life in a way that even I had not anticipated, and it's very, very exciting. Now, all of this is just information from one tissue section. To date, we've collected at least 552 such tissue sections with almost 3 million individual spot level measurements. Wouldn't it be exciting if we could combine all of that information to see much higher resolution at what's happening in this these types of tissue sections? How does this change depending on which cell types there are? How does this change with age? What does it mean for health and disease? So that's where I'm really excited to be able to partner with this team and see where these kinds of visualizations will take us and how we can get a deeper appreciation for what the data are telling us about the underlying biology of senescence.